Hello students, welcome back to my channel. Today I will discuss about genetics, some basic fundamentals from genetics for class 10 ICSC, CBSC and West Bengal Board of Secondary Students. Especially from genetics, there are Mendelian inheritance and laws from the Mendelian inheritance. So I am starting from the previous class. In my previous class, I told you in our body number of chromosomes 46. So we have 46 chromosomes, means 23rd pairs of chromosomes are present in our body. Among 46 chromosomes, 44 autosomes are present and 2 sex chromosomes are present. Now, how can you segregate that chromosome, or how can you identify where is the location of that chromosome? So, if you see the structure of nucleus, within the nucleus chromatin materials are present, and from chromatin materials chromosomes are comes out, looks like that. So these are the structure of chromosome we are getting from chromatin material from nucleus. So 46 chromosomes are present in our body. Among 46 chromosomes, 44 are autosomes and 2 sex chromosomes are present. So in male body, 2 sex chromosome is X and Y, but in female body, 2 sex chromosome X present. So during transmission from the parental character towards the offspring, so if father will give only X chromosome, then the mother will give X, then the product will be daughter. If father will give Y chromosome and the mother will give X, because no alternation is there, so there will be son. So sex determinating factor ultimately depend on father, because in mother 2 sex chromosomes are present and they both are X chromosome, but in father 2 sex chromosomes are present, one is X, other one is Y. So if father will give X chromosome, then there will be daughter. If father will give Y chromosome, there will be son. So depend on father, sex determining factor. Similarly, among 46, as there are 46 chromosomes are present there, so these are 23rd pairs of chromosome. So 46 chromosomes, these are in pair. If you think one pair, second pair, third pair, so the pairs are looks like that. So in first pair, 2 pairs of chromosomes are there. Similarly, in second pair, 2 pairs of chromosomes are there. So in each pair there are 2 chromosomes. Look at the second pair. Here 2 chromosomes are present and they both are equal in their size. These kind of chromosomes are known as homologous chromosome. And here see the structure, these are not same. 2 chromosomes are completely separated in their structure. So here where the structures are present and 2 chromosomes are completely same according to their shape, size, these kind of chromosomes known as homologous chromosome. So it is very important for Mendelian experiment. So I am starting from monohybrid cross, then I will explain about dihybrid cross. Now what is monohybrid cross and what about dihybrid cross, that I will explain now. Monohybrid cross means Mendel has taken to do this experiment one tall or pure tall plant, especially taken pea plant, that is the scientific name Pisum sativum. So Mendel has taken pea plant and the plant that is pure tall, and the plant which is taken, it is not pure tall, it is pure dwarf, means short height, and it is pure character. So 2 pure characters are present. How many characters are present here? 1 pair of character. It is tall, it is pure dwarf. So 2 character means 1 pair of character is taken, contrasting character, and doing that experiment. That's why the name of it known as monohybrid cross of Mendelian experiment. So 1 pairs of character, pure tall, pure dwarf, and after that it is parental generation. That's why I have written here P generation. From parental generation when it is transmitting towards the offspring, that is known as F1 generation. And in F1 generation, look at the structure in F1 generation, that is capital T small t. So 50% character from pure tall plant and 50% character from pure dwarf plant. But if you see the plant, it will be a hybrid tall. So where is the character of pure dwarf? Because it is pure tall, it is pure dwarf, so it will be mix up character, 50% character, but ultimately it is hybrid tall. So here the characteristic features of dwarf plant is ultimately suppressing. So it is dominating character which is suppressing the character of pure dwarf plant. So this type of characteristic features here, it is dominating character, it is recessive character. Which one is dominating character? Pure tall is dominating character. Why? Because pure tall is suppressing the characteristic features of pure dwarf. So capital T, small t, after that one T from here, one T from here, and there will be capital T, small t for F1 generation. That is F1 generation, and it is hybrid tall. Now F2 generation. What about F2 generation? Look at here. In F2 generation, capital T, small t means the product of F1 generation, they are crossing. That is self-fertilization. When they are crossing in F2 generation, they will form the gametes capital T, capital T, capital T, small t, capital T, small t, small t, small t. Why? Because if you see the checkerboard is there, which is capital T, small t and capital T, small t. If you see, these are male gametes, these are female gametes. So capital T, small t, these are male gametes, these are female gametes, and they are producing capital T, capital T, capital T, capital T, capital T, small t, capital T, small t. Also capital T, small t, small t, small t. So 4 offsprings are there, and look at the character. Capital T, that is pure tall, small t, pure dwarf, but capital T, small t, that is hybrid tall. These kind of characteristic features we are getting. So ultimately, the dwarf plant, the character that is recessive, it will not lost. We are getting the character. It is 25%. If you see the total characteristic features, that is 100%. So ultimately we are getting the characteristic features from here: 20% pure tall, 20% pure dwarf, and 50% hybrid tall. Look at the structure, 50% hybrid tall. So why it is 50%? Because it is 25, it is also 25. So 25, 50% character. Now what is phenotype and what is genotype? If you see anything externally, means we say the man is tall, the man is dwarf, so it is very short height. So externally, if you see any characteristic features, that is phenotypic character. And if you want to see the characteristic features from internally by the analysis of their genetical structure, that is genotypic character. So in capital T from externally, that is pure tall. Similarly, capital T, small t, if you see externally, that is also, you think that is pure tall, but ultimately it is not pure tall. But if you see externally, that is pure tall, but not internally. So we are observing the character externally. That's why phenotypic character, in phenotype, mind it, in phenotype, capital T, pure tall. Similarly, if you see capital T, small t, you will think that that is also pure tall. So they are under tall group. So capital T, 25%, capital T, small t, say 50%. So total 75% tall characters. And short height dwarf character, that is 25%. That's why phenotypic character is 3:1, means 75 is to 1. Sorry, 75 is to 25, that is 3:1. But if you think the genotypic character, that is different. Why? In genotype, capital T, pure tall, but capital T, small t, externally you are not thinking about it. You have to think according to genetical analysis. So that is capital T small t. So it is not pure tall, it is impure tall, or you can say hybrid tall. So this is also another character, and another character remaining here, small t, that is dwarf plant. So how many characters? Three characters are there for the analysis of their genotype, because genetically character in genotype, if you want to analyze, then capital T one character, capital T small t another character, small t another character. So capital T 25%, capital T small t 50%, and small t 25%. So that is 1:2:1. So after Mendelian one monohybrid cross, what is the phenotyping ratio? That is 3:1. And what is the genotyping ratio? That is 1:2:1. So that is the Mendelian cross related to monohybrid cross. You can take here any other characteristic features, just like capital B, small b, and if you think that these are pure black, and if you take any other character just like in pure black, so you can do that experiment with the help of Mendelian experiment. Now I will discuss about dihybrid cross in next topic.